Students, let us start with amino acids. By now, you know amino acids are formed by the degradation or breakdown of proteins. They are the primary units of proteins. Now, these amino acids are chemically composed of carbon, hydrogen, oxygen, nitrogen, and to some extent sulfur and phosphorus. The structure of amino acid can be simply studied like this. See, we have a central carbon atom. To this carbon atom, one of the valency of carbon is taken up by a carboxylic acid group, COOH. So, this is the acid group in amino acid. Another valency is taken by an amine group, NH2 group. Third valency is taken up by hydrogen. And fourth, there is an alkyl chain and hydrocarbon chain, which if is only H forms the glycine. If this R is represented by CH3, it is alanine, and if this R is represented by CH2OH, it's serine. Like this, R can be any hydrocarbon chain, but it is the amine group and the carboxylic group, and definitely the R chain, which gives the characteristic property to these amino acids. Now, these amino acids are of three types, depending upon the number of amine group and carboxylic group. These are the acidic amino acid, like the glutamic acid. It can be basic amino acid like the lysine, or it can be a neutral amino acid like valine. Now, since the carboxylic acid group is just next to the carbon, or the carbon as the same carbon atom has on one end the carboxylic group and the other end amino group, this is known as alpha amino acid. That is, the same carbon is having the carboxylic group as well as the amine group. Such amino acids are known as alpha amino acids, and all the living organisms contain this alpha amino acid. There are 20 types of amino acid found in the living organisms. Some of these amino acids are synthesized within the living organisms from amination, deamination of other amino acids, and these are known as non-essential amino acids. There are near about say 13 to 11 non-essential amino acids. Why 11? I'll say later. There are 13 to 11 non-essential amino acids. And the essential amino acids are those amino acids which are not synthesized in the living organisms. They are to be supplied from outside, like valine, leucine, isoleucine, lysine, phenylalanine, tryptophan, tyrosine, and methionine. These are all essential amino acids. Now, in some books, there's a third category which is termed as semi-essential amino acid, because the rate of synthesis of these semi-essential amino acid is really slow. So it's better if they are supplied from outside, like your arginine and histidine. Threonine is an essential amino acid for humans, but in other animals they can synthesize threonine in their body. That is why we say there are 7 essential amino acids in animal and 8 essential amino acids in human. The rest 12 in some, according to some scientists, are non-essential amino acids. Then histidine and arginine for some are semi-essential amino acids. Now, these amino acids, they have the typical properties. Like the amino acids, they react with acids and bases together depending upon the pH of the medium, and hence are amphoteric molecules because they react with acids as well as bases. Now, these amino acids, when dissolved in water, they exist as zwitterine. Zwitterine means carrying a positive and a negative charge together in a molecule. See this structure of amino acid. Here, the carboxylic acid group is the hydrogen from the carboxylic acid group is dissociated into COO minus and H plus. This H plus goes into the medium, forms hydronium ion, and the hydronium ion again donates its hydrogen ion to the amine group, forming NH3 plus molecule or NH3 plus group. So see, in the same amino acid we find a negative charge as well as a positive charge. So this is the Zwitter ion configuration of amino acid, where the amino acid is carrying a positive as well as a negative charge. Now students, as I said, these amino acids are the building blocks of the proteins. They join with each other. These 20 amino acids join with each other to form long chains. And just the arrangement of amino acid forms the primary structure of the protein. It is actually not the final protein. Then the coiling and supercoiling of the polypeptide chain occurs to form the active protein. So we can say proteins are the heteropolymers of amino acids. Why heteropolymer? Because it consists of different amino acids and not a homopolymer. Now, it is the peptide bond. It is the peptide bond which actually joins the different amino acid. And how is this peptide bond formed? This peptide bond is formed between the carboxylic acid group of one amino acid and the amine group of another amino acid, with an elimination of water molecule. So what do I mean by that? Suppose this is my one amino acid and I have another amino acid here. Let me write it in this way. It will be easier for you. Whatever R group may be there. But if there is an elimination of water molecule from the OH group of the carboxylic acid and hydrogen from the amine group, a bond is formed between the carbon and nitrogen of two molecules. and this is what is known as the peptide bond. So how will we represent that? So water has been eliminated. So let me write it in this way. This is the peptide bond, which actually joins the two amino acid, and this way a long chain of amino acid is formed. Then coiling, supercoiling takes place to form the functional protein. Now these proteins are the essential components of, you can say, the cell membranes, the cell organelles. They form the basic structural cytoskeleton of the cell. They are required for various hormones, enzymes and everything. Proteins are, in fact, they are omnipresent. They are present everywhere and they are needed everywhere.